You're listening to Antiviral Frontiers with your host, Rick Kiessig. In this episode, we're speaking with Professor John Coffin of Tufts University School of Medicine about his more than 50-year study of endogenous retroviruses.
Hello, and welcome to Antiviral Frontiers. I'm Rick, and like to introduce yourself?
Yeah, I'm John Coffin. I'm a professor of molecular biology and microbiology at Tufts University in Boston, Massachusetts. And I have been working on retroviruses for more than 50 years, I hate to say.
Wow, impressive. Yeah, it's I'd be interested in hearing a little bit about your current work.
Okay, our current work is actually in two areas. One has to do with effects of HIV on its infected host. We're particularly interested in a rare but very interesting mechanism by which the virus may occasionally cause cancer in a very direct way. And we can talk about that in more detail, of course. And the second is an interest of ours that goes back quite a long ways before HIV was discovered in around 1980. And that is endogenous viruses, which are fossils of retroviruses that infected our very distant ancestors. And because they were retroviruses, and a few still are actually, we can read the fossil record in a very interesting way because we can infer the property. Retroviruses, as your audience I'm sure well knows, have the unique property that their genome is copied into DNA, and that DNA is regularly integrated into the genome of the host cell, so they become a part of the genetic information of the host cell.
Right.
And because the retroviruses have been infecting our ancestors for literally hundreds of millions of years, possibly even more than that, we can read that fossil record to learn about ancient pandemics, if you like, or retrovirus infections.
Right.
Where, to some extent, where they came from, and importantly, how and why they died out, because there are no, well, with the exception of HIV, which is brand new, and HLV, which is not quite so new, but newer. None of these others, and we carry many of them that are extinct species that are no longer extant at all. They've died out completely. And one of the things we're interested in is why they might. We think we found a mechanism that's important for their dying out.
So is HIV, is it not permissive to germ cells? Is that why it doesn't enter in as ERVs?
HIV requires a receptor that's not found on germline cells.
I see.
Unlike most retroviruses that we have studied for many years, use sort of housekeeping proteins that are expressed really on all cells, things like receptors for uptake of amino acids, and things that all cells have to do. And that those are the more common retrovirus receptors are proteins like that. But HIV uses requires two proteins to enter, both of which are really quite specific for cells of the immune system and a few others, but not many. And so that really restricts how the cells they replicate in, unlike the other retroviruses we've studied for years.
Interesting. So here at ChimerMed, we're developing a family of broad-spectrum antiviral therapeutics, and so this whole area is really quite fascinating and interesting to us, ERVs in particular. I understand that ERVs are associated with cancers in some cases. Is that correct?
There's some mouse models where ERVs can, and which we studied quite intensively back in the 80s, particularly, and that where the viruses are, unlike the viruses in humans, some of them are the ERVs in humans. Some of them are quite recently integrated. By recent, I mean within the last few 100,000 years, or even less. and are still capable of encoding infectious virus. None of the ones we have.
Really?
We have maybe 80,000 of these bits and pieces of DNA in our genomes
That many?
that are derived from retroviruses,
Mm-hmm.
and some of them can code intact functional virus proteins. Very few of them, but none of them is capable of encoding a complete infectious virus. So the mechanism that you see in mice, where you see activation of some of these quiescent endogenous proviruses during early development in the animal, and then spread of the virus. The virus is made through the animal again, and then causing cancer in the same way as a virus infecting from outside would. That doesn't happen in humans.
Okay. So you get virus-like particle formation
or
virions, or
in some cases you can get virus-like particle formation.
Okay.
And again, in humans, in no case are they complete and infectious, but they can look pretty good in the EM. There are only a few of the HERVs, so-called the human endogenous retroviruses, that are capable of encoding intact-looking virions. And a very large fraction of them actually have they share the same mutation in the envelope gene, which creates a frame shift early on, does not allow it to be expressed, actually also probably also damaging to the pollen reading frame.
Does that mean that they never transition to like a lytic replication mode?
That's right.
There's no like dsRNA formation or VRC formation or anything like that?
No, there's nothing happens with those. It's possible. Yeah. It's possible on paper to align the two genomes and draw a recombinant between them that's infectious, and people have actually done that. Have taken a collection of the most recent of these. Some of these are very recent, and we can use the properties of retroviruses to infer unusual things about these that we could not do for normal cellular genes, by and large, or for other kinds of viruses that you might see fragments of in the DNA, which do occur. We can take for example, the fact that you have these long terminal repeats at the ends of the DNA form, the provirus, the integrated form. The fact that those, from the way they're made, if you draw out the replication scheme, you see that the way they're made requires that the two long terminal repeats be absolutely identical in sequence at the time that they're made.
Okay.
But with a long time evolution, the two LTRs will independently accumulate mutations and differ from one another. And that creates a very, very nice clock you can use. It's a very slow clock. It takes a few 100,000 years before you see a single mutation, given the slow germline mutation rate.
Okay.
But in things in the millions of years scale, you can use that very nicely to date when that integration occurred.
Wow, that's cool.
And that's a tool that we've used. We and other endogenous retrovirus people have used quite a bit.
So I think I remember.
Just we call ourselves.
Reading somewhere, my memory on this is a little fuzzy, but that something about like in cancer cells that are exposed to excessive stress, like say from radiation, for example, that they can express HERVs sometimes? Is that like there's some increase in HERV formation in cancer cells?
Remember, in the first place, there are a lot of them.
Yeah.
In really all animal genomes are really loaded with the HERVs, with HERVs of various kinds. The ERVs are under two kinds of control. One is from things that are outside. If they're sitting down in a gene and they can become, the expression of that ERV can be under the control of the gene that they're sitting in. That's one thing that can happen. But the thing people think of most is that the ERVs carry their own LTR sequences, and LTRs are just full of promoters. So that's a promoter that is tuned to in the virus that it came from is tuned to assist the lifestyle of that particular virus. So different retroviruses will tend to replicate in different cell types. They'll have different ways of transmitting themselves from one individual to another. And this is all dependent on their being able to express themselves specifically in certain types of cells. And with retroviruses in general, it's probably better for them to be fairly specific because it does not do a virus any good to cause too much disease. To cause any more disease, it needs to do to help its own transmission.
Yeah.
And so retroviruses different retroviruses tend to have very specific target cells that they go after. And a lot of that target cell specificity is mediated. In HIV, it's all partly mediated by the receptor usage. But in many retroviruses, I said, they use very broadly based receptors. And so their specificity is more dictated by the specific sequences in the LTR, which couple it to specific developmental stages and specific cell types that is useful to a particular virus for its own purposes, and its own purposes being to make more of itself and get transmitted to another individual.
Yeah, we've tried. We did some experiments where we use lentiviruses to make stable cell lines, and one of the things that we encountered in that process was just the high risk of the DNA insertion causing cancer in those cell lines. So I would imagine that would be a problem in humans as well, right? How do they? Is there some mechanism that retroviruses use to minimize or prevent cancer formation? Or
being as specific as they possibly can is certainly part of that.
In terms of cell type or in terms of where they are and where they insert in the DNA?
There are mouse models that let you see that very nicely. There's a virus of mice that's been studied a lot called mouse mammary tumor virus, which causes a mammary carcinoma, breast cancer, if there is such a thing in mice.
Right.
That virus is actually transmitted through milk from mother to offspring.
Okay.
And it has a very complicated life cycle in the baby it gets into, where it goes from one cell type to another cell type, and finally to the cell type that's actually going to become the mammary secretory mammary cell, and mouse milk is just loaded from an infected animal. It's just loaded with these virus particles, but they're not seen in most other cell types.
Okay.
But there people have isolated mutants of this virus that can cause T cell lymphomas, for example. And so those mice would die before they have much time. It takes a while, of course, for a virus like that to be transmitted because it goes from mother to offspring, and so the baby has to get infected and then grow up to become enough to become a mother again in order for the virus to thrive and live on.
All right.
It's important for that virus not to cause any unnecessary disease, even though eventually that mother will die after due to the kind of insertional mutagenesis you just mentioned, of the virus happening to integrate next to one or another proto-oncogene, we call them, or sometimes just called cancer genes.
Yeah.
I hate that term.
Right.
It implies that their real function is to cause cancer, which is not the case. There are genes that can be abused to cause cancer, but it's abuse, not the natural lifestyle that causes that.
Makes me wonder about the differences between retroviruses and herpesviruses and the way that they do their DNA integration. I gather that herpesviruses are just not permissive to germlines, and that's why they don't spread that way. Or
there is one that spreads as an endogenous virus, HHV6, I think. That actually many of us carry as an endogenous virus.
Oh, really?
And it's an infectious virus, but that's a very rare example.
Yeah.
Probably a one-off kind of thing that just happened to get fixed in a lot of people.
Do you do much work with herpes viruses in your lab?
But I've got a lot of colleagues that do. I don't work with herpes viruses at all.
Yeah.
too complicated. I mean, they're too big.
Right.
Uh,
Yeah. Interesting. It's yeah. Um, is there immune system activation with
there can
herbs or
there can be, um,
innate immune system? I mean.
not necessarily, but it can happen.
Uh-huh.
And an interesting feature is that many of them have LTR sequences, that for whatever reason, but probably relates to the lifestyle of the original virus that they came from.
Hmm.
Are many of them have promoter sequences that are sensitive to interferon, uh,
Okay.
being turned on by interferon induction, and so quite a number of interferon responsive genes in humans have been shown to be that because they have one of these responsive LTRs sitting next to it, and that's what's actually turning on that gene expression. And that is probably, in the cases where that happens, it's probably something that actually has been selectively advantageous to have that gene come under interferon control.
So is this like related to a cause of chronic fatigue or something like that?
Yeah. If you want to get into the cause of chronic fatigue, that's another story altogether. But
Yeah. Just curious if there's a connection between retroviruses and chronic fatigue. We
there
have an interest in it from
was at one
the
time.
EBV side and
There was at one
herpes
time about
viruses.
15 years ago, but thanks to some of our work and some of our colleagues, that went away very quickly.
Okay.
If you want, I have a review in a recent annual review of virology that you might be interested in reading on this. It's a kind of a fun story in a way.
Oh, okay. I'll check that out.
Fatigue is not a fun thing. I mean, it's a real disease, and
Yeah.
it's almost certainly the same as long COVID, and it's probably may well be caused by one of these sort of non-pathogenic coronaviruses that have been around for a long time.
Right. Yeah, I mean, there's this collection of viruses. I don't know how, I'm not sure if this is the case with retroviruses. Maybe you would know, but certainly with the herpes virus family, there's, and papillomaviruses. I mean, some of these are like present in 80% or something of the population.
Yeah.
So, and obviously the herbs are there in all
Yeah.
of us.
Yeah, right. I mean, Epstein-Barr virus is even more than that. It's in the 90s somewhere.
Right. Yeah.
And and the HERVs, they can't be, grossly pathogenic because that is the simplest way that a HERV goes away. Of course, is it just never gets fixed because it's so pathogenic that it affects the reproductive capacity of the individual it's in. That individual never becomes a parent.
Right.
May never even grow up, or may never even fully develop. So lots of HERVs are in. And when you look at the distribution of HERVs in the human genome, and this is true if you look at any animal, you'll find that the older they are, the less likely they are to be in genes. Only the very recent ones are still in genes, and those are, and even a lot of those are still very selectively in genes, because they tend to be. Well, it's not strictly true. They tend to be, even if they're in an essential gene, they tend to be lethal only if the individual is homozygous. And that requires, after a while, the overall population effect will be enough of homozygosity, which that all depends now on the population structure and interbreeding and the inbreeding coefficients and things like that.
I see.
But that will be enough eventually to weed out such things by selection if they're basically recessive in that way.
Interesting.
So they will go away, but they'll go away more slowly than
Right.
ones that have dominant effects like causing cancer should be weeded out fairly quickly.
Right.
as far as their integration site is concerned. And the reason that we have these cancer models in mice is not that the mice have actually survived having these for long periods of time, but that they have been spotted by curious scientists, and so their selection was that curious scientists gave them a good home and bred them in. There's a strain of, just to get off topic even more, there's a strain of mice that we used to study that we discovered a mutation that was caused by integration of a retrovirus within a gene that's necessary for hair formation.
Okay.
And so these mice actually, the baby hair comes in, but once the first growth cycle of hair is required, it all falls out and nothing more, no new hair grows in. And this was discovered by a mouse fancier. There's still groups of people who their big hobby is breeding this and that mice for this and that property.
Yeah.
So these mouse fanciers happened to be night watchmen in the aviary in England, and saw this mouse with no hair go running across the floor. Caught it and was able to actually breed the gene for what we call the hairless gene, HRS. Breed it into inbred mice, and that mouse is actually quite widely used in Australia. And it's used to test sunblocks on, which is, of course, a big deal in Australia, having good sunscreen.
Yeah. Interesting, yeah.
Hard to test on a mouse because of all that hair.
Right, sure.
If you breed a mouse with no hair, then so anyway.
Yeah.
Actually, we were actually able to get very large, have individual a collaborator looking at very large numbers of mice looking for revertants in that mouse that lost its hair, because we'd shown that mutation was due to the insertion of a retrovirus, and sure enough, the revertant that had no hair, that retrovirus had gone away.
Interesting. Do you happen to know if it's in general possible for not a nerve, but let's say for a conventional retrovirus like lentivirus or HIV, that to force it to transition from latent replication to lytic mode?
A lot of people are interested in that question.
Yeah.
And it's a huge topic in HIV research, with the idea that since HIV is largely a lytic virus, if you can take these latent viruses, take cells that we know exist in very small numbers, very small proportions. The numbers are actually fairly large, but the proportion is very small. Mm-hmm. I mean, we have 10 to the 14th CD4 T cells, so it doesn't take a very big proportion of that to be a lot of cells.
Yeah.
A very small fraction of those will carry these latent HIV genomes in people on therapy.
Right.
Unfortunately, a much larger fraction carry defective dead HIV genomes that make experimentation very difficult, because they put a lot of noise into your experiments when you're trying to see when you're trying to look for these kinds of induction events and things.
Right.
And so far, those strategies have not met with really any success whatsoever of trying to turn on all these latent proviruses, and nobody really understands what makes them latent. The best model that I know of is that it's sort of in a sense a transcriptional noise kind of model, because of the factors that HIV needs for its own TAT and Rev, and that it needs for its own expression. They can sort of turn on and off, and if they turn off, they're sort of self-promoting. If they turn off completely, there's no good way to turn them on again. And so that may be one of the basic mechanisms of silencing is sort of that, in a sense, noise model.
So if they stay turned off, are they still a health hazard? Or?
They never stay. Yes, because they never stay turned off forever.
Oh, I see. Yeah.
a latent provirus, by definition,
Yeah.
a latent provirus is one that is capable of reactivating,
Right.
and probably does so at some point. And we and others have shown over and over again that these pools of latent proviruses, the cell occasionally turns on and emits a burst of viruses, and nobody completely understands it. Nobody really understands exactly what's going on there.
Interesting.
The importance of integration factors, of integration site, for example, and controlling that, the importance of the viral factors, the importance of genetic variation in the virus, in the provirus itself, that can affect whether this happens or not, and so on.
Yeah.
And it's a very, it's been, it's a topic where I like to say that the better the science, the worse the news.
Right.
The more you learn, the harder the problem becomes.
Yeah. Yeah, I can see that.
You need the information, but you may not like the information that you get as far as your goal of trying to come up with a cure for HIV infection, because obviously it's the resurrection of HIV after you stop therapy that is the real target in trying to develop a real curative strategy, particularly given all the failure of all the vaccine efforts.
Yeah. That's, I think, definitely an area of interest for us, for sure. I've also been curious about whether some aspect of HERVs could be used either as an indicator, let's say, that something is malfunctioning in certain cells or cell types, or
people work on
signal
that
for therapy in some way.
Yeah, people so far, nothing's really been reduced to practice. But the idea that, as you mentioned at the beginning, that in some cancers will express HERVs that the normal equivalent cell does not. Maybe perhaps under some special circumstances, but just because there's a difference. Cancer is a disease of gene expression, of course, and proviruses are the products of gene expression, and so changes in gene expression could easily change the product. It could easily be expected to change the patterns of provirus gene expression as a side effect, and that indeed happens, and there are many cases of that, and people have at least considered and worked a bit on trying to develop targeted therapies against viral antigens. One has to be a little careful there because these proviruses that are expressed, that may seem to be specifically expressed in a tumor cell relative to the normal cell that the tumor comes from, may in fact be normally expressed in some other cell type somewhere in the body that you don't know of, and so you might get some rather unpleasant side effects if you go in with a strategy to kill specifically those cells that express this antigen. You may find all of a sudden that some important normal cell type is also expressing it that you don't expect. You can't test every cell type. And you can't just test tissues, because the tissues are mixtures of cell types. And if you see virus expression, you got to figure out which cell type it's coming from.
Right.
So it gets more complicated.
So with the high mutation rates that we see in cancer cells, are the ERV parts of DNA particularly prone to that sort of mutation in some way?
Not obviously. Not obviously. They're no more so, I would say, than any other piece of random piece of cell DNA. That is to say, the mutation rate is not very high. If you put the right kinds of selection on it, but cancer cells tend to often have altered mutation rates anyway.
Yeah, exactly.
And that's in in many cases, that's probably what got them to be cancer cells in the first place. Is they were for whatever reason more prone to mutation than other cells. And so the same would be true of the HERVs that the cell carried.
Yeah.
Some types of cancer cells are quite impressive, actually. Teratocarcinomas, which are, as you probably know, as your audience probably knows, are cells of the germline. They grow these tumors that have oddball bits of organs in them, like teeth and things like that.
All right.
And but they also there are a couple of proviruses that they express at a high level that are actually have intact gag genes and are capable of making particles. They're not infectious, but they can make dramatic amounts of particles.
Oh, really?
And it's because of these specific proviruses that just happen to be are probably ones. Yeah, I would propose that the proviruses they express are ones that were normally expressed in that cell type.
Yeah.
And that's an example of the sort of thing that saying, so this would be cells, some kind of very early developmental stage where they would be expressed and then probably turned off again later on normally. And so treatment with an antibody or some other strategy that specifically went after that particular HERV might have dramatic effects on pregnancy, for example. Might also cause abortion at an early phase and things like that. So, and that would be the kind of side effect that I would be concerned about. That might be.
So can HERVs, does that imply HERVs can have adaptive immune system impacts then as well as innate?
Well, certainly in principle. In practice, I can't think of any examples off the top of my head where that's true, but it certainly could, yeah.
Interesting. One of the themes of our podcast here is underappreciated aspects of viruses.
Okay.
There's a lot of really wild and strange things in the virus world. I'm curious what sorts of things in you and your
case of HERVs, one of the things that we think is underappreciated, even to many virologists, is their importance in evolution as part of the host virus arms race. And in a sense, when you think about it, the most dramatic property of all of the 80,000 or so HERVs that we carry is that, without exception, they're all extinct. These are fossils of viruses that have gone extinct.
Okay, right.
And a big part of our research program is devoted to looking at one of these proviruses in particular, which we hypothesize, it would be a difficult thing to prove absolutely, but I think the evidence is going to be pretty good that this may be the case, that this one provirus was the cause of extinction of this particular species that it belongs to.
Really?
This one provirus we discovered, and I should say one of the nice things about studying HERVs and other endogenous retroviruses is that because they're retroviruses, you can use principles of retrovirology to really aid the paleontology that you do to try to figure out what the life cycle and lifestyles and function of this virus actually was and how these events occurred.
Okay.
And so we can postulate, we can provide good evidence, I think, for a very rather complicated evolutionary history of this virus, this particular provirus with its host, going back 10 million years.
Wow.
This is a virus that integrated first into the genome of an ancestor of both humans and gorillas.
Okay.
And already your retroviral insight comes into play because you realize that once a virus integrates into a specific place, it's there. It doesn't move around.
Yeah.
And it may create new ones by integrating elsewhere, but that one will still be there, or its residue will still be there. And so if you see a provirus integrated in exactly the same place in humans and gorillas, it means it has to have been integrated into a common ancestor, been present in a common ancestor of those two animals.
Sure.
The last common ancestor of humans and gorillas was between 8 and 12 million years ago. So we just pick the middle and say 10 million years.
Right. Yep.
And so we know that virus, that's from a virus that was extant 10 million years ago. We also know that there's another related but distinct provirus in our cells as well, that is almost exactly the same for a region of about, I don't know, a third of the genome maybe, that extends from the middle of the GAG gene to into the Pol gene, into the gene encoding reverse transcriptase. The GAG gene encodes the structural proteins of the virus.
Okay.
And that sequence is identical, almost virtually identical. There's a very few differences that have accumulated over time, over the 10 million years since that. We know that that's a recombination event that must have occurred 10 million years ago, because that same recombinant is found in all the other, in gorillas, humans, and chimpanzees as well.
Wow.
And so it almost certainly occurred at the time that provirus was made. It was made by the way retroviruses recombine is by putting two different genomes in the same virion, and then when reverse transcription occurs, it jumps back and forth from one genome to the other, which reverse transcriptase does a lot anyway.
Right.
So that must have happened with this particular virus 10 million years ago. And then it integrated into this, this site on chromosome 3, where we now have it to look at. The GAG gene has remained completely intact in all of these host species for that 10 million years. And so, a lot of mutations occurred in 10 million years, and for it not to have accumulated any significant mutation in all that time is really quite remarkable.
Yeah.
It certainly implies that it had some important function. But when it picked up the GAG gene from this other virus, it also picked up a mutation, which is a single base deletion in one portion of the capsid protein, the protein that codes for the internal structural proteins.
Okay.
And that one base deletion causes the capsid not to be able to form its structure properly.
Okay.
And if that's expressed at the same time as a good virus, the proteins will mix, and the mixed protein will also be unable to form a proper structure. They look quite ugly in an electron micrograph when you see them. And if we want to go back still further, the virus that was the donor of this oddball GAG capsid gene was a closely related virus, but replicated in a somewhat different way. They're actually, retroviruses can replicate. There are two sort of different modes by which new viruses can be made. The standard classical retrovirus mode is the GAG proteins assemble at the cell surface and then gather the genome with it and wrap the membrane around it and bud off. And these are the pictures that everybody knows well with HIV, where you're seeing the bloody virus coming up.
Sure.
But there's another mechanism which some viruses use, particularly some viruses of this group, and that is they assemble the capsid structure is assembled in the cytoplasm, free from the membrane, and then eventually finds its way to the membrane and wraps it around.
Mm-hmm.
And it turns out this mutant GAG protein, the one that has this one amino acid deletion, belonged originally to a virus that assembled in this way. And that's because of differences in another part of the GAG gene that are the other side of the site of recombination.
Okay.
That contains the amino acid sequences that direct the GAG protein to the membrane.
Mm-hmm.
So this other type of virus, they don't go to the membrane; they just remain in the cytoplasm and assemble there, and then they find their way to the membrane somehow and put it from there.
So do I recall correctly that some HERVs have been shown to be involved in placental formation?
That's right.
Or something about
That's actually a very striking feature. That's a process that is referred to as exaptation, which is the use, the taking of the co-option of endogenous provirus for some valuable thing for the host.
Right.
There are probably a lot of examples of exaptation. I mentioned the LTR sequences that have put some genes under interferon control. So that's an example of exaptation of the promoter sequences. And there are also exaptation of protein sequences. And this incitins which is the family of proteins that you mentioned. And that's very dramatic because every mammalian cell line, except one, has active incitins that were derived in this way. And they were derived from different original viruses. So the ones in primates, and all the primates have the same, have two actually, syncytin 1 and syncytin 2, and they and they have the same function. They do it slightly differently, so that it actually leads to slight differences in the way placentas are formed between mammals and rodents, and that's correlated with the nature of the virus that actually became a syncytin in these species. But it happened over and over again, and all probably, and so I speculate, again, evolutionary speculation that you can't really prove, but speculate, given its persistence, its reappearance in all of these different, humans have, primates really all have one set of them, ungulates have another set, rodents another, and so on and so forth.
Okay.
The only exception here is marsupials.
Okay.
Now, what the syncytin does is it causes the external cells of the placenta of the which is encoded by the embryo or fetus, causes them to fuse into a syncytium, hence syncytin. And what that does is removes all the spaces and gaps between cells.
Okay.
That would allow the diffusion of small molecules, bad small molecules from the maternal circulation to the fetal circulation. It sort of ensures that the only thing that can get from the mother to the fetus is things that are actively recognized and pumped through.
Yeah.
Using specific mechanisms, things that it greatly limits the ability of things to get through passively.
Right.
From one next. And as you might expect, if you make it in mice, this has been done, I think in some other species as well. But if you knock out syncytium, hence syncytin, what you find is that gestation becomes very short. It simply can't survive well enough. I mean, structurally it looks okay, but presumably they're just things that the mother's eaten that the developing embryo can't tolerate, the developing fetus can't tolerate. And that shortens the gestation period. And I think you could argue that being sort of basal to the roots of all mammalian orders, except marsupials, these syncytins actually are what allowed mammals to become more than just little bitty things that run around, allow them to develop for much longer periods of time, become much more complex, grow big brains rather than just little bitty things that can run around to get stomped on by dinosaurs.
Right.
And may in fact underlie all important mammalian evolution, if you like. May in fact have been allowed. That would not be the only gene that would be necessary for that, but it would certainly be one that was critical.
So these incitins are the gene.
They're all endogenous.
It's a ERV.
It's an ERV, and if you look at the sequence of particularly humans, I think in the others, although I haven't really seen data, but I would expect it's the same. If you line up that sequence against the closest related exogenous virus you can find, you can see that the envelope gene, which is what becomes the syncytin, of course, it's a cell surface protein that's encoded by the virus, is completely intact. The other genes are just trashed. They've just accumulated mutation after mutation after mutations. They're obviously no good at all.
Yeah.
In at least the human case, you can take that syncytin gene and you can put it into a virus, and that virus will still work, despite the fact that that gene has been in our DNA and that of our ancestors for 50 million years or more.
Wow.
And so that clearly tells you that all of the functionality of that gene, the ability to interact with the receptor, the ability, and the specific receptor it interacts with, the ability to fuse cells, which is what the envelope gene does for the virus, has been preserved over all that time. Whereas genes that aren't doing anything for it have just become, have been abraded by the sands of time, as I like to say.
Right. Do any of these ERV genes from these 80,000 viruses, do they encode reverse transcriptase?
There are a couple in the most recent group, which is a group that we work and most people work on actually, called ERVK HML2 or just HML2. It's not the derivation, that's not important. Yeah. There are a couple of them that can encode intact reverse transcriptase. And in fact, the structure of one of those was just published a couple of years. It's remarkably like HIV for some reason, although the sequence doesn't look like HIV.
Does the presence of that indicate health condition or anything?
That one doesn't seem to have any particular properties that we know of with regard to human health or any correlation with anything interesting. And I'm not even sure that there are a couple of them, and I'm not even sure they're capable of being expressed except in the laboratory.
So they're not innate immune system triggers or anything?
That I'd have to go back and check. But,
yeah.
So it's just an oddity as far as I can tell right now. Now somebody may come up with something that changes it. Will change everybody's mind on that issue. It's entirely possible. It happens a lot. So,
do ERVs emit any sort of like nucleic acid material into the cytoplasm that could get the innate immune system excited, or do they pretty much stay in the nucleus? And
people hypothesize that retroviruses are very carefully designed to avoid tickling the innate immune system.
Okay.
And people have really only, it's been obvious that it had to be true for a very long time, but the science has only caught up to what I thought obvious to begin with.
Yeah.
That sometimes happens, and sometimes things I thought obvious to begin with turn out to be completely wrong. But that's not in this case.
Yeah.
And what has to happen when the virus comes in, what the cell really dislikes, and what the innate immune system goes crazy for, is DNA in the cytoplasm.
Right.
And also certain kinds of RNA, but particularly DNA.
Yeah.
The kinds of RNA, the virus has pretty carefully evolved its RNA so that it looks enough like cellular RNA that it doesn't trigger alarm systems, because that has to get out into the cytoplasm to be expressed as messenger RNA, and on its way to making virions and so on. DNA is another story. So what has to be true, and this is something if you look at old textbooks, they show you something that cannot be true for just this reason. What has to be true is that the virus, when the DNA is being made, and for a long time people thought the DNA was being actually made in the cytoplasm, which is still partly true. I mean, the DNA synthesis starts in the cytoplasm, but it's now, in the case of HIV at least, it's now known to not be completed usually until after the structure gets into the nucleus. But in order to do so, the DNA has to be within the capsid structure, which has to be very tight. And if you infect cells at a very high multiplicity infection, lots and lots of virus particles, you put a lot of virus so that each cell takes up lots of virus particles. some of those virus particles, particularly if you mishandle it in the lab at all, some of those virus particles are going to have capsids that have been broken and so on, and they'll start to do reverse transcription, and then you get a good innate immune response. So very often, the virus you grow in cell culture infects cells at a high multiplicity will give rise to a good innate immune response.
Right.
However, if you infect cells so that only one cell, only if where the multiplicity is less than one virion per cell, then you often won't see that, and that's because the virus, the cell that can replicate the virus is not the one that has the innate immune system activated, and that's because the capsid structure is so tight. And people didn't appreciate this because they didn't completely understand the capsid structure, and until quite in the last few years, it's really quite recent. And then there's a requirement for a specific cellular small molecule in the cell that kind of locks the capsid up. And if you run assays in test tubes without that molecule, then the capsids fall apart. So you think the capsid will have really soft structure, but once you have this key in there that locks it up, then the capsids are actually quite tight, and that's all been shown not by us, but by other very good.
So during HIV viral replication, it produces dsRNA at some stage, presumably, with viral replication compartments?
Not as a regular thing, no. That's another thing that it would try to avoid.
Okay.
Quite a bit. The DNA, the RNA in the virion as it comes in is as a dimer whose structure is still not very well understood, but it almost has to be true that it has short double-stranded regions all along, just by chance, when there are chance sequences that are nearby on the genome where one is the inverse complement of the other for maybe half dozen bases or so. Those will make a little base pair stretch there, and that'll hold it. That's almost certainly what holds the DNA together. But it's also got a protein on it called the nucleocapsid protein that helps it to that mediates the ability to form these base pairs and so on.
This is a common feature of all retroviruses, then, that they don't really form dsRNA.
Yeah. All retroviruses have this, but that's locked. That again locked within the capsid.
Yeah.
It's not accessible to the signaling mechanisms that would activate the innate immune response for the same reason that the DNA is in it when it gets made.
Okay. So they don't form viral replication compartments like most viruses, then?
No, it doesn't do that. No. Oh. No, it's retroviruses. They all have in common very similar structures. Overall, there are a number of differences with specific proteins that some have and some don't. But the overall structures and fundamental mechanisms are pretty much all the same.
Is there some similarities here with hepatitis B?
There is and there isn't. Hepatitis B uses a reverse transcriptase, but it does so in a sense rather differently. For one thing, in the case of hepatitis B, reverse transcription occurs before the virus, the particle is released. So what's in the genome is actually the DNA,
right?
In the virion. Whereas RNA with retroviruses, that doesn't happen until after the virus gets into the next cell.
Right.
Hepatitis B does not integrate in any regular way. You can get in hepatocellular carcinomas, you can get integrated hepatitis B DNA, but it's a much more random event. It's much more like what happens with, say, papillomaviruses, where you have all of this DNA around, and just like what happens when you transfect a cell with DNA, all this DNA around, some of it by more or less random mechanisms gets incorporated into the cell DNA, but it's not in any regular way.
So higher cancer risk then.
There's no specific enzyme responsible for that the way there is in retroviruses.
Yeah.
So and it's not a requirement for the replication of these viruses. It's very much a side effect. It can be for pathogenesis, it can be an important side effect, hepatocellular carcinoma in HPV-infected people. But
Yeah.
as far as the virus is concerned, it's off target.
Yeah. Okay. Well, I think that's probably it for today. Thank you very much for your time. Really appreciate it. It's been great chatting with you. I learned a lot, and very interesting field.
If anybody has any questions, I'd be happy to answer an email.
Yeah, sure. How should we listeners reach you or get a hold of you or your?
They can reach me, john.coffin at tufts.edu. That's easy to remember.
Awesome. Thank you very much.
We hope you enjoyed this episode of Antiviral Frontiers. To hear more from the fascinating world of viruses, biotech, and medicine, please visit our website or find us on Spotify and YouTube and follow us to get future episodes.